Good evening. I hope my voice is audible. Can anyone confirm if my voice is audible? Okay, perfect. Thank you. So today's session we will be discussing about designs of clinical trial. So I have created some designs of clinical trials wherein we will be discussing about how the clinical trials best can be designed. So the first topic which I'll be taking is presence and absence of control group. The clinical trial designs can be classified into uncontrolled trials and controlled trials, which I'll be discussing further in this times. And the second classification based on participation allocation. So this classification, it can be randomized and non-randomized. If it is randomized, stratified randomized, blocked randomized, and also cluster randomized. So there are three different types of randomized also: the cluster randomized, block randomized, and also stratified randomized. Then I'll be speaking about the classification of randomized clinical trials into parallel study and crossover studies. Then I'll be discussing about the awareness of the treatment drug, whether it is open label or blinded. If it is blinded, single blind, double blind, and triple blind. Then I'll be speaking about based on significance, it's a bioequivalence or it's a non-inferiority studies. So these are the classification which I'll be discussing for today's designs of clinical trial. So the first one is on presence and absence of the control group, uncontrolled study or controlled study. So controlled clinical trials or uncontrolled clinical trials. The second one is randomized clinical trials. So one is non-randomized, the second one is randomized. In randomized clinical trials also, I'll be classifying subgroups: stratified randomized, blocked randomized, and also your cluster randomized. Then I'll be discussing about randomized clinical trials, parallel study, and crossover study. Then I'll be speaking about awareness, open label, blinded, single blind, double blind, and triple blind. Then I'll be speaking about the based on significance, which is on bioequivalence or non-inferiority. So without any delay, let's go into the designs of clinical trials. So every clinical trials varies on the designs based on various factors. These factors will be on the primary clinical research question, the phase of trial where you are conducting which phase one or two or 2A or 2B or 3A or 3B, based on therapeutic area or indication or disease characteristics, which disease you are doing it, based on the targeted patient population. So these are the factors which has to be taken into consideration when you are designing a clinical trial. So the clinical trial will be designed based on the following factors. The first one is based on your primary clinical research question, phase of trial. Third one, therapeutic area or the disease characteristics. And fourth one is based on target patient population. The first classification of the designs of trial will be based on presence and absence of control group, based on presence and absence of the control group. First one is uncontrolled trials. Here, the efficacy and toxicity of medication is being compared with a group of patients without having a control group. So only one group will be there, and this is not being compared with a control group. Either it is placebo or treatment group, you are not going to compare it. Those kind of trials are called as uncontrolled trials. The efficacy and toxicity of medication is compared with a group of patients without having a control group. Then it is called as uncontrolled trials. So opposite to this is controlled trials. So basically, uncontrolled trials are being conducted in phase 0 and phase 1, most of times for tolerability and also for safety. And sometimes in phase 2 also you will do uncontrolled trials. Most of times it's a controlled trials which are being done. So what is controlled trials? Opposite word of uncontrolled is the study is being compared with a control group, which may receive a placebo or treatment drug or a comparative drug or effective treatment. So both of the group are studied simultaneously. One group is being given your test drug, another group is being given your uh, control group, which is your placebo or another effective treatment. So now these are called as control trials. So there are two types of trials based on the absence of control group. One is uncontrolled, where you have absence of control group. One is controlled group, where you have comparing with active control or your placebo control. So which is called as controlled clinical trials. So furthermore, the clinical trials are being classified or designs are being further classified based on participation allocation, how the subjects are being allocated to treatment drug. So this is more important topic because most of times you end up having a bias in clinical trials. How can you avoid the bias? How can you continue with having the clinical trials without having a bias is something which we are going to discuss now. So based on the method for the participation allocation, there are two methods. One is non-randomized and one is randomized. Non-randomized investigator who allocates the participants to different treatment groups. The investigator itself allocates the treatment groups to the subjects. Then it is called as non-randomized. But randomized, opposite to this word, is participation or randomly assigned to the treatment groups or arms or groups by using closed envelopes or computer generated or system generated sequence or random numbers. So randomization is avoiding for bias. So there are two types: non-randomized, where investigator gives your treatment group. One more is non-randomized or randomized, where automatically by using element of chance, either system or closed envelope systems are being used for providing your treatment groups. Then it is called as randomized. Randomized trials are being used for preventing bias. So first thing in the randomized is stratified randomized. So what is stratified is grouping a particular variables for subjects. Like for example, male subjects who have not taken a drug under the age of 18 group age is called as stratified. So one particular variable is being given. So strata are constructed based on values and variables, and randomized scheme is implemented separately for each stratum. So first you will create a groups, stratas, stratums, then based on that particular stratums you are going to implement randomization. So this is what you call it as stratified randomized. So based on this, you need to have objective of the stratification should be make sure that each number of stratification have same number of subjects. So to avoid strata with less number of patients, we need to make sure that strata should be very, very minimal. So the classification of stratification should be very much minimal. So the grouping of subjects based on variables or the data points should be very minimal. The next one is block randomization. So the block randomization is the method designed to randomize the subjects into groups that results equal amount of sample size. Each block will have same number of subjects, and in that you will go and put your randomization. Though this method ensures that balance of sample size across group over the time. So blocks are being created to make sure that the same sample size is being done across all the blocks. And cluster randomization. The third one is cluster randomization. The interventions is administered to a clusters by randomization to provide or to prevent the contamination. Each clusters forms a unit of trial. Either it is active control or comparative. So one cluster is being given active control, one cluster is being given the placebo control, so that it avoids or it prevents your contamination across the system. So this is what you call it as cluster randomization. So we have discussed uncontrolled studies, which is being directly given by investigator, and then the controlled studies where it is comparing with your active drug or treatment drugs. And then we started discussing about non-randomized, where investigator will assign the subjects, randomized where system will assign the subjects, then stratified randomized, then blocked randomized, and then cluster randomized. The one more thing is randomized clinical trials are being classified into two more types. One is parallel group and one is the crossover group. So parallel arm design is most commonly used designs when you have two treatments to be given, A treatment and B treatment. The both the A treatment and B treatment are not being interchanged or not being exchanged between the subjects. The same subject is taking same drug, then it is called as parallel group. Say for example, group A is taking only test drug, group B is taking a reference drug, then it is called as parallel group trial design. So there is no exchange of the treatment in between the trial, then it is called as parallel group. But crossover, it's a two treatments which gets exchanged between A and B sequence. Say for example, group A is taking test drug, group B is taking reference drug. The next visit, group A will take reference drug and group B will take test drug, which is called as test and reference, reference and test. So which is called as crossover. Parallel studies are without exchange of the drug, interchange of the drug. Crossovers or interchange of the drugs. And next one is we are going to discuss about the classification based on the investigator awareness of the groups which are being assigned. So which is called as, first one is open label, wherein open label, the subject, investigator and sponsor, all the people will be informed about the treatment drug. All the parties are informed about the drug, then it is called as open label trial. Investigator is aware what groups is taking what drug, and even subjects will be aware that what treatment is being given to the subjects. Then it is blinded studies. One or more parties are kept unaware, either it is treatment drug or placebo, or active drug or placebo drug. And here you will have zero bias or less amount of bias when you take it as blinded study. So blinded are different types: single blind, double blind and triple blind. Single blind, you have participants do not have or subjects do not know about who is taking what drug. But double blind is the subject and investigator both are unaware of treatment drug. So both are unaware of treatment drug. Then it is called as double blind. Most of trials are being conducted in double blind trials. Triple blind is indicating the investigator, patients, and as well as sponsor are unaware, but there will be an unblinded statistician who will assess the unblinded statistician. So based on the awareness of the drug, it's an open label where everyone knows about the drug, where blinded study, which is classified into single, double and triple. Single blind, only subject is unaware; double blind, subject, investigator is unaware; triple blind, subject, investigator and sponsor is unaware. So now we will take a quick recap. First one is based on control and uncontrolled studies, based on randomized, randomized and non-randomized, block randomized and stratified randomized and cluster randomized, and then randomized clinical trials, parallel study and crossover study. Then you have open label, single blind, double blind and triple blind. And last one is based on the significance of the results which is found between two groups. So first one is equivalent trials. The equivalent trials is the studies are intended to show the test drug is equivalent to reference drug. So equivalence trial is the studies are intended to show test drug and reference drug are same or having same efficacy. Then it is called as equivalent trial. But non-inferiority trials are the studies intended to show the investigational drug is not worse than control drug. So there is a test drug and reference drug or control drug. So test drug is not worse than your control drug. Then it is called as non-inferiority trials. So there are two types of trials. One is equivalence trial and one is non-inferiority trial. Equivalence trial is when you say that your test drug and comparator drug or control drug are same, then it is called as equivalence trial. But when you say that non-inferiority is when the study included to show that the investigational drug is not worse than control drug. So now today's topic, the designs of trials. The initial topic I started with how the factors are included in designs of clinical trials. The first one is the different factors which are including is primary research question, clinical research question, phase of trial, and also therapeutic area or disease characteristics and the patient's characteristics or target population. So these are the factors to assess the clinical trial designs. The clinical trial designs are following the uncontrolled trial where there is no comparison of controlled study, the controlled trials where there is a comparison with controlled trials, and there is a method of participation of allocation which is non-randomized where investigator will directly allocate the subjects to the treatment drug which contains some bias. Randomized clinical trials which element of chance, we are using the system based element of chance to assign the subjects where you might not have a lot of bias. Stratified randomized where you will go for a particular variable and you will stratify the subjects. In that stratification you will randomize the subjects. And then you have a block randomization, the block randomization method to design the randomly assigning the subjects to equal sample size in each group. So then it is called as block randomization. Cluster randomization intervention is administered to clusters to prevent contamination. The parallel studies is same treatment is being given across the visits. Crossover study is interchange of the treatment across the visits. And based on the availability or awareness of the treatment drug is open label, wherein all the three parties are aware of the treatment drug, and blinded study wherein one or more parties are unaware of the treatment drug, and single blind where subject is unaware, double blind where subject and as well as investigator is unaware, triple blind subject, investigator and sponsor is unaware. And last one is based on the significance where one is equivalence and one is the non-inferiority trial. Significance is where the test drug and control treatments are made equivalent. We are assuming that both are equal. Non-inferiority trial is studies intended to show that investigational drug is not worse than control treatment. So I think today's topic I have covered most of things, but I will take one or two questions from the group so that I can answer the questions. So I hope the session was useful, and we have discussed about uncontrolled trials, controlled trials, non-randomized trials, randomized trials, stratified trials, blocked randomized trials, and cluster randomized trial, randomized clinical trial, parallel study, crossover study, open label studies, blinded studies, single blind, double blind, triple blind. Based on significance, one is bioequivalence and non-inferiority studies. So I think I'll take one question here: how to determine the appropriate sample size for clinical trial. So this question, most of times you should understand that before we take the appropriate sample size for any clinical trials, we will do a thorough pilot studies to understand what is the power, statistical considerations, and different variables for disease or drug or all those characters. Then only you will come to know about your sample size. So basically, you need to do a power analysis to estimate the sample size. So statistically, you will conduct the sample size calculation by statistical methods, considering the factors. What are the factors is anticipated the effect of size, variability of outcomes, the different variables of outcomes, alpha errors, beta errors, and the level of confidence. So ANOVA, the analysis of variance or level of confidence is being included to understand the number of subjects to be taken into your clinical trials. Most of times we will conduct a pilot study to understand this level of confidence, ANOVA, and also variables, and also outcomes and alpha and beta errors, and all those aspects will be taken into consideration for understanding the sample size. The sample size is being done in order to make the study in proper way, and the study should get clear. So I think I have covered most of points today. The designs of clinical trial, factors affecting designs of clinical trial, uncontrolled trials, controlled trials, randomized trial, non-randomized trials. In randomized trials, I have discussed about stratified randomized and block randomized and cluster randomized, and randomized parallel study and randomized crossover study, open label, blinded study, single blind, double blind, and triple blind, and based on significance, which is bioequivalence and also non-inferiority. I'll close my session today, and I'll meet for one more live session next week. Thank you so much.